Thank you very much. So unfortunately for some, I'm not going to be talking about REM, the band, but I am going to be talking about the Relational Event Model, abbreviated to REM, to investigate non-human behaviour. And this is work co-authored by me with Chris Markham, who's based at the National Institutes of Health here in the USA, Blake Morton, who's based in Scotland, in Stirling, with Hannah Buchanan-Smith's group in psychology. I don't know if anyone knows them. And also my mate Selv de Court, who is based at Manchester Metropolitan University in the UK, just down the road from me. So Blake and Selv provided the data on the corvids and the monkeys that we're going to look at. And Chris has been very important in data prep and modelling, assistance with the modelling. So we've worked as a team, and I'm delighted to have the invitation to this meeting and the opportunity to talk about this. So I thought I'd have a slide about what I'm not talking about and then talk about that. First of all, I've just had a paper published in JRSSA, the Journal of the Royal Statistical Society, which is it's got a lovely endearing title of Multiple Membership, Multiple Classification Models for Social Network and Group Dependencies, which I've learned to be able to say in one breath. And what I think that the important bits are in the green box there. What do I think this is useful for non-human behaviour? Well, if you've got some kind of individual, I keep calling it Y variable, but some kind of individual measure on the nodes of a network that you want to look at variations in, then that could be something like fitness, although I'm probably too naive to know what sorts of measures you would want to use in animal behaviour or non-human behaviour. But let's say it's fitness, then a perfectly reasonable question might be to ask, where's the variation? Is it by the individual, given the age of the non-human or the gender or whatever? Is it by the network that they hang out in? Is it by habitat? Potentially, we could also think about stability through time, but the important thing also at the end then, it could be a relative share of variation down to habitat, network, individual characteristics. And so knowing about the relative share of variation in something that we can measure on the nodes of a network, I think has got potential, and I'd be interested in talking to anyone about that might be interested. And then a multi-level version of that, or a multiplex. So if you had more than one network for the non-humans that you wanted to compare in terms of variation, there's a potential way of doing that for a paper I've just submitted to Social Networks for a special edition for which I am co-editor, but I will not be reviewing it someone else is reviewing it and editing that one. You'd be pleased to know. But I think the relative importance here would be on variations where you've got networks of non-humans, guppies or zebras or whatever it might be, but you've also got networks of habitats, and that could be something like a river system or something like that. So that was me talking about what I'm not going to talk about, and now I'll talk about what I am going to talk about, which is to demonstrate the research value of the REM, or the Relational Event Model, for studying non-human behaviour. And there's a very important publication by Carter Butts in Sociological Methodology from 2008 that I would urge you to look at if you're interested in this kind of thing. So I'm going to outline the REM just very briefly in one slide. Carter's paper is a much better place to find out the expanded story, but I'll outline it on one slide and illustrate it with two examples. One for corvids, so food sharing in jackdaws, and one for monkeys, in particular the tufted Capuchin monkey. And I'm going to describe the examples and, very importantly, I think, discuss the importance of taking into account the data collection and the setting. So I'm always telling my students that, yeah, you can take survey data and put it into SPSS, get some results as if it was a simple random sample, but the computer won't complain, but your results don't make sense, the standard errors will be too small, this kind of thing. And so we need to take into account the way in which the data were collected when we're analysing it, and sometimes the way it was taken, the way it was sampled is actually interesting as well, so that's a bonus. So how do we take into account the way the data were collected? How do we take into account what can happen at any point in time in our model? Then I'm going to summarize the results and then just make some general conclusions about where I think we could go with the REM in animal or non-human behavior. So one slide of algebra then, the background to the REM. We define, this is where I could do with a long stick or a pointer, but never mind, define relational event tuples as A is the set of things that involves I, J, K, and T. So I is the sender of event A, J is the receiver, and oh, is this going to work? Oh, wonderful, thank you. So I is the sender, J is the receiver, K is the action type or category, and T is the time of the event. And so, without going into too much detail, we can look at the dynamics of this using a rate function involving this lambda function here. Of all of these things I've just talked about, the sender, the receiver, the action, and then you've got some covariates potentially, and also you can look at the, well, we take the past action into account when thinking about the current action, and then theta we can think of as some coefficients that we're going to estimate. And so the effects that we can consider would be fixed effects for each non-human in our network. So if we had 12 jackdaws, then we can have a fixed effect for each of those. We can look at persistence of action. So one jackdaw might persist in trying to give food to another one that perhaps it wants to mate with or something, and so there's persistent action. We can look at a P-shift effect where jackdaw A gives something to jackdaw B, and that results in B giving something to C, so there's a kind of transitivity. And we can think about reciprocity, which of course means that an action from I to J is followed at some point by an action back from J to I. And what's interesting is we can think about exact time data here. So if you've videoed something or you've recorded it very precisely, there's the potential to put that exact timing information in the model. But also we can work with ordered events, which is quite often the case that with our data where we know the order of something but not the exact time. And so adapting the argument of Goffman from 1967 to animal behavior, actions amongst animals over time can be seen as a series of discrete events where one animal directs a behavior at one or more of the other animals in their environment. So we can think of things like feeding, grooming, or aggression as being actions from one animal to another. But of course, there could also be actions that are directed to themselves. The most obvious one perhaps is eating a food item rather than sharing it with another animal or non-human. And so I should say that what I'm going to do here with the examples are on ordered events rather than exact times, just due to the nature of the data that I've looked at. I'm using the R package rel event, which was developed by Carter Butts, again, to go along with his paper on the methods. And like is often the case in social network analysis, certainly for modeling, data prep takes a lot of effort to get it right, to take into account all the things that need to be considered, like data collection and so on. And so there's a very nice package in R called InformR, which was developed by Chris Markham, one of the co-authors of this work. And I think the reference for that is 2011, but if you go onto the R website, you can find it. And support constraints. This is a very important thing, that these models allow you to specify at any point what actions could actually take place, or, if you like, which actions you can observe given the nature of your data collection. So a bit like in football or soccer, if the referee's looking at something, then the referee can decide if that's a foul, but if something's going on away from the ball, then that's not necessarily observed, but it would still constitute a foul if it had been seen, but we didn't, well, the referee didn't see it. In an animal or non-human networks, if you're focusing on a particular monkey and some other monkeys are grooming one another, you might not observe that, but it doesn't mean it's not happening. But then we need to take into account what we can actually see in our data collection. So first example then, European jackdaw, Corvus monodula, so one of the corvid family that is among the more intelligent birds. And so we're interested in the behaviour of these. And I just thought I'd start with a couple of aggregated networks for this. So these are the time aggregated information that I got for the 12 jackdaws, colour-coded here by family group. So these jackdaws, the data were collected when they were in an aviary, but they were originally from particular nests, and the colour codes tell you which nests they were from. And this is a particular food item, pieces of sweet corn, I believe, which are edible to jackdaws, and they're going to eat them, but they're not particularly high value. If we then look at the, and sorry, these lines are representing, these arcs are representing the transfer of items. So the majority of the time, the birds just eat the item, but some of the time they don't. If we then look at waxworms, which are a kind of luxury food item, I believe, for these corvids, then there's much more transfer going on, and particularly where birds are from the same nest. So we can see that these edges are actually, or these arcs are actually weighted by numbers. So we can see, for example, here a large number of transfers of waxworms. So we can see that given the different food items, there's different amounts of transfer. And we can also see that there are within nest, there's a suggestion of within nest transfers being higher than between them. But what we can't see is the rate of persistence or reciprocity. We can't see how these actions follow one another to see if there's any kind of interesting pattern there. And if we just look at this static network, we're not really taking into account data collection and this kind of thing. So it looks nice, it's a good starting point, but it's really then to show you what you can do with the disaggregated afterwards. So this particular example for the jackdaws then, 12 jackdaws received 28 feeding trials between the 25th of June and 8th of August 2003, so a while ago now, and they were in an aviary. One bird at a time received 10 food items consecutively, after which the second bird received them, and so on until they all had received the same treatment. And these were randomized in each, for each day, and also alternated between sweet corn on day 1, waxworm on day 2, sweet corn day 3, waxworm day 4, and so on. And this waxworm I'm talking about is Galleria melanella, if anyone knows about them and would want to know more. So the birds didn't receive any sweet corn or waxworms outside the trials, the feeding trials. And so you can think of the experimenter choosing a jackdaw at random, offering that jackdaw a food item. So if it's jackdaw 1, the jackdaw can either eat the item, decide to transfer it to any of the other 11, or I think the phrase, is it consented theft, where you could sort of allow someone else to steal it. So some kind of transfer could go on. So for each food item provided to the focal bird, we recorded whether the bird ate the item, transferred it to another bird, or dropped it on the floor. And the transfers were divided into two categories: recipient initiated and donor initiated. So if it's recipient initiated, that's when the recipient took the food item directly from the beak of the donor, or when the donor put the food item on the floor and the recipient took the item. But if it just dropped the item and walked away, then that wasn't a transfer. And donor initiated was when the donor put the food item directly into the beak of the recipient. So there's different kinds of transfer. We can also look at whether donor-initiated transfer were preceded by begging behaviour by the recipient. So there's lots of information recorded, and the original analysis of the aggregate data that I showed you partly on those sociograms was that juvenile jackdaws shared food regularly with more than one individual, and donors initiated a large proportion of transfers. That jackdaws reduced the frequency of food sharing and number of recipients, so there was more sort of focused sharing during the 2 months of study. Jackdaws shared significantly more highly preferred food than less preferred food, so they were more likely to share these waxworms than the sweet corn. And the results were consistent with two of the proposed explanations for food sharing, reciprocity and harassment avoidance, but we couldn't really look at these tests for persistent for things like reciprocity with this. This was a static analysis. So what the REM can do is to look at things like reciprocity, the frequency of reciprocity, and also persistence, whether a particular bird tries to do something over time and how frequently they do that. So these were based on time-aggregated data. The REM is going to be used on disaggregated events data. And it turned out that really it was only just over 1/10 of the food items that were shared by jackdaws rather than eaten by them. And there wasn't much evidence of more food sharing between sexes. So when we put gender in as a covariate for the REM, there wasn't much evidence. But that's probably because only 2 of the jackdaws in the sample were actually female birds. So there's a biased sex ratio in the data. The way that we do this in the REM for the jackdaws is each morsel, we follow the path of the morsel, if you like, as it enters the system and until it leaves the system, so until it's eaten by a bird or dropped on the floor. So we treat the introduction of a morsel, so giving the experimenter giving the jackdaw a food item as an exogenous event, randomized by trial and bird, that statistically and in reality we can think as initiating an event history. And once the bird receives the morsel, 3 things can happen. It can eat it, it can transfer it, or it can drop it on the floor. So when I'm talking about these action types, as I was talking in the model specification, the 3 action types there are transfer, eat, or drop. We're going to use an ordinal likelihood because we know the order of events but not the exact times. And we're going to use support constraints that take into account the setting and data collection and so on. So we're going to look at 4 types of behavioural effects. First of all, the behaviour of individual birds in terms of each morsel of food. Secondly, whether homophily of birds from the same nest is associated with particular behaviour. Thirdly, whether reciprocity and persistence of actions occur. And fourthly, is there any difference in behaviour for different food types? So the first thing we did was fit a model with 13 fixed effects. You've got 12 jackdaws. Each of them has a turn in being offered the food. Therefore, each of them has the chance to eat or transfer a food item. And we've also got the possibility of dropping the item on the floor. So we've got 13 fixed effects with eating the morsel as a reference category. And we look at each bird and see what actually happens in that case. Positive coefficient suggests that a bird's more likely to give a morsel away than to drop it on the floor than to eat it. Negative coefficients indicate the bird's more likely to eat the morsel. So we can take into account these things for the food type, for whether it's a bird from the same nest, and so on. So some birds we know were raised in common nests, and we can look at the tendency for birds of a feather to flock together by including one homophily statistic to the model that compares the likelihood of an exchange between birds in common nests to those that are not raised in the same nest. And so if we get a positive coefficient for homophily, we're saying there's more likelihood of exchange of food items for 2 birds from the same nest than 2 each from a different nest. And obviously a negative would indicate the contrary. To investigate reciprocity, we add 2 statistics to the model. First one is just reciprocity in general, and the second one is the recency of that, whether it's a recent reciprocity of an event or further down the track of the event history. And again, just like in regression, generally a positive coefficient means more, and a negative would be less. And so we can see whether there is any evidence of greater reciprocity or lesser according to which combination of characteristics we're looking at. We also look at persistence in the model by adding a statistic that captures overall tendency for a bird to repeat its last action. So persistence, again, can be added. And because we know that certain types of food may be more desirable, in other words, waxworms are more desirable than sweet corn in this case, we can take that into account with a covariate as to say whether it's sweet corn or waxworm. And so we did this with these different types of model, and we evaluated the model using the Bayesian information criterion in each case. And the negative fixed effects coefficient from M1, which was the fixed effects only model, suggested that basically that birds prefer to eat the food rather than to transfer it or drop it. However, there was some evidence that jackdaws raised in the same nest are more likely to share than those raised apart. So if you take the coefficient for something, excuse me, and take the exponential of it, it gives you a relative hazard. So we can say that there's sort of 2.36 times more likely to share between homophilous pairs, so birds from the same nest, than one from two different nests, or one each from a different nest. And we also found quite a lot of persistent of action. I decided not to put loads of results in really small font that you wouldn't be able to read or remember, so I'm just telling you the summary of the results. We also found that a hazard that a bird will repeat whatever he or she just did was e to the 2.26, which was basically a tenfold increase. So there's a very good likelihood that the last thing that the bird did is alter the next thing. We found very little evidence of reciprocity, but when we did find any, it tended to be right away rather than further along the chain of events. That's something you would never be able to determine with an aggregated data set. So here we can say, well, how quickly was there a reciprocal action, if there was any, and it tended to be quite soon. We found that the homophily effect disappears in the presence of persistence and recency effects, suggesting that there's an interaction of homophily and those effects. So what that basically suggests is that reciprocity is confined to the nest. And finally, we found that evidence that waxworms are particularly valued as a social commodity. In other words, there's more likelihood of sharing those than just eating them. Because this was shown from the coefficient of the covariate for that. So having corn also drives birds to persist in their actions to a greater extent, and there was less evidence of reciprocity for sweet corn than for waxworms, which is what it says on that slide, just those last 2 points there. Okay, so there's an example with the corvids. Let's look at another example for capuchin monkeys, in particular the tufted capuchin. And here, these are interesting because of their behavioral and cognitive complexity and striking similarities to chimpanzees in terms of personality structure, social learning and tool use, sharing habits, cooperative behavior, and so on. So like other primates, capuchins engage in reciprocal acts, food sharing, grooming, support during fights. But a recent study, apparently by Anderson in the Journal of Cognition, that some of you may know this, I haven't actually seen this, but I was told about this just before I got on the plane, that there's this particular point, so I need to read this one. But it suggests that capuchins recognize third-party exchanges like humans. And so results of that study indicate that cognitive or emotional prerequisites for judging reciprocity in third-party social exchanges exist, but in at least one other primate species. That might be true in other primates, but it's unclear due to lack of data. I'm sure there are people in this room that know more about this than I do, so you might want to comment on that. But the patterns of reciprocity of interest are of interest for primatologists because they relate to why or how primates cooperate, which helps address evolutionary questions like altruism and intelligence and so on. So how can the REM help us learn more about these longitudinal patterns? Well, static analyses don't give us the full picture, because animals base their current and future interactions on prior experience, And so we can't see that from a static network. Having said that, oh, it's not the next slide, but I will show you the static networks as a starting point. But I'll just tell you the situation, the setting for these data. So we've got 2 enclosures here in Edinburgh Zoo in Scotland, an east group with 8 monkeys and a west group with 10 monkeys. Now, by design, east and west cannot interact. So a monkey in the east enclosure cannot groom a monkey in the west enclosure and vice versa. But of course, with only 18 observations, you want to get the power out of the data, and really you'd like to combine them in the study. So can we do that, and can we take into account the focal sampling that was used here to collect the data? Well, the answer is yes, but let's just first of all look at the static networks just to give you an idea. So this is the east group with 8 monkeys, Chico, Junon, Popeye, Kato, and so on. And this is the weighted arcs for grooming. So we can see evidence of reciprocity here and so on over, time aggregated. And here is, yeah.
When you have reciprocal exchanges, it looks like they're equal weight both directions, or is that just the combined weight of total time spent grooming either direction?
I think that the way it's been drawn is the combined, but I take your point. Yeah, it does look equal. You could make these arrowheads proportional if, yeah. But I think it's been done equally, but with the sum of the, yeah. But I know what you mean, yeah. But anyway, the point is that we're not going to stop at the static network anyway, but thank you for that. And we got 10 monkeys in this west enclosure, 8 of which interact, and actually 2 of whom, Pedra and Toka, don't interact in terms of grooming, certainly at the time that this study was observed. So we've got 18 event histories on the sending and receiving of grooming actions between pairs of monkey. And these data are egocentric in that each period of observation, and thus each event history, follows a focal monkey for a fixed period of time. And for that monkey, we can say who that focal monkey groomed, who groomed the focal monkey, and we can keep this for an event history. And we've got these 2 separate enclosures, so what we want to do is to pool the data so we don't lose power, rather than dealing with a group of 8 and a group of 10 separately. So we can use a pooled egocentric relational events model with an ordinal likelihood. And what we do is then use support constraints to take into account actions that can't or can occur. So if it's an east monkey and a west monkey, we use a constraint to say that that's not possible that one could groom the other because of the nature of the setting. And we take into account the focal sampling, again using these support constraints. What actions can you actually observe at a particular event point? And this ensures that the correct set of possible events are used in the model estimation. And so we use fixed effects of grooming and being groomed. So there's 2 lots of fixed effects. One is for a monkey to groom another, and the other one is to be groomed by. And we were interested in particular in reciprocity of grooming and persistence. And so we calculate these different sufficient statistics, or we specify these in the model. The reciprocity one sums the number of times grooming by one monkey is reciprocated by another, and then we've got the number of times a particular monkey continues to either send or receive grooming, given that they've just groomed or been groomed respectively. And again, just quickly from the results, we found significant variation in the sending and receiving of grooming across focal actors. That is, that some monkeys were more involved in grooming than others. Well, we can see that in some cases there was no grooming action going on at all, but there certainly was also variation in those that did groom and were groomed in the rates of that. When we add reciprocity, then evaluating by the BIC, the Bayesian information criterion, we get a great improvement over the basic model, which strongly supports the idea that monkeys reciprocate these grooming actions. So we find that the hazard of returning the favour, given that one has been groomed by a particular monkey, is multiplied threefold. So if we take the exponential of that reciprocity coefficient, we get 3.28. So this is 3 times more likely that, this is the hazard. And the evidence from Model 3 supported the notion that monkeys really enjoy their grooming behaviour. So they're likely to continue receiving grooming from a particular monkey once it's begun. And we find that the model that includes all of the effects for grooming and being groomed and persistence is the best one. And we could go on and add gender effects, so we could look at homophily and heterophily of gender in terms of grooming. And we could combine the persistence of being groomed and grooming statistics into one to simplify the model. Sometimes, just like any statistical model, there's the trade-off between parsimony or simplicity and what you can realistically look at with your data and what you would want to find out about. And so there's the tables of coefficients that I'm not going to repeat talking about, you'll be pleased to know, but just to prove that I did them. And there's another one. But let's just conclude now that, that the REM is a useful way of studying animal behaviour or non-human behaviour over time. For example, persistence or reciprocity of action. And we could allow for a single action, which is grooming, or for multiple actions, such as sharing, dropping, or eating food. And we note that one of those actions is a loop, a self-action, eating an item, oneself, as it were. So we can include loops in there. Data collection and setting can and should be taken into account in the REM specification. And this could include the focal sampling that's used, or impossible actions because of different enclosures and so on, or impossible interactions. This works with exact timed or ordered data, and a multi-level version has recently been proposed which would allow, if you had lots of groups of event histories, that's another way. So instead of having 2 enclosures, if I had 20, then I could think about a multi-level version of this to see if there's a common set of behaviour that is common to all of the enclosures. So that's another possibility. And I'd just like to say thank you for listening, and thank you for inviting me. If you want to email me about this, there's my email. And I'm based in Manchester at the Mitchell Centre, so that anyone that's in the UK, we do, and wants to know more about SNA, we do short courses and summer schools and so on. So if anybody is going that way and wants to know more that's not so familiar with SNA, this might be useful. When my website works, that one, otherwise the university one has also got some information. Thank you very much.